And first of all, before we embark on our talk, we thought we'd share with you a short film which illustrates some of the energy and enthusiasm of the DEEP project. We'll set up from here.
Welcome back to DEEP, the Dornoch Environmental Enhancement Project. We're out on the Dornoch Firth, heading towards the oyster beds, where we're going to start to lay the first reef in over 100 years. In the next few months, 20,000 oysters are going to be down there living happily in the Dornoch Firth. It's a great day for us, so let's go!
So I'm Dr. Bill Sanderson, I'm from Heriot Watt University, and I'm the Science Director of the DEEP project. So we've done the historical research, we did the survivability study, and now it's time for a much larger deployment into the Dornoch Firth. First we had to work with our partners to find enough European oysters, and then we had to look at the biosecurity aspects. So now we're putting in a much greater number of oysters, ramping up to 20,000 initially, then 200,000 oysters, and then 1 million a year until we get to 4 million. First, we've got to create the habitat that's long been lost. So first thing in the morning at 4 AM, we loaded the new habitat, the shell material at Invergordon. And then the barge brought the material around to the Dornoch Firth and started to lay the new habitat for the oysters to live on. Whilst that was happening, the actual oysters that had been through an extensive biosecurity process were sorted and taken to another vessel for deployment. Then the dive started. The Firth is a challenging environment to work in, strong currents, difficult visibility, but we got the shell material down and we got the oysters down onto the habitat. We could even see some of the oysters opening up and starting to adjust their new environment.
Science and innovation underpins what DEEP is all about. And this is led by the Heriot Watt team.
So my research with DEEP allows me to measure calcification, respiration and the filtration of shellfish in real time and getting carbon values for this. And this in itself allows us to value the restoration of a reef.
My research with DEEP has tried to understand what makes larvae settle. So that's the babies of the oysters when they metamorphose to an oyster that will stay in that location permanently. And the main thing we found is that the larvae prefer to settle on their own species. This means that for restoration efforts, the larger the oyster population, the more successful settlement will be.
Personally, it's fantastic to get the project going here. I mean, the partnership is a genuine partnership between Glenmorangie, Heriot Watt and the Marine Conservation Society, and because of that everybody's pulling in the same direction and it really does work.
So that's it, another stage complete for DEEP. It's been a fantastic team effort with lots of people involved, and we're very proud to be pioneering native oyster restoration in Europe.
Okay, thank you. So some of you might have seen that before, but it's captures some of the energy and enthusiasm of the project. And "Let's go" is Hamish's one-liner from that, which stays with me. And many of you might be thinking, what is a whiskey company, Glenmorangie, doing involved in an environmental restoration project with Heriot Watt and the Marine Conservation Society? And I'll give a little bit of background to this before we press on into the main part of the presentation. So next slide, please. So Glenmorangie very much champion the idea of sustainability in the work they do. And this strapline of theirs uses the word Tain in the middle of sustainable, which of course is, Tain is where their distillery is up in the Highlands. Next slide, please. And sustainability for them is really about future-proofing their history. They pride themselves on having been on the banks of the Dornoch Firth for 176 years. And they plan to be there for another 176 years. So they're not a sleepy, old-fashioned company. They're very innovative as well, and they look to their environmental credentials as part of the core of their business. In 2017, they introduced a new, very innovative anaerobic digestion plant, which is highly efficient. And I'll come back to talk about that in a bit more detail later. Next slide, please. And as a consequence of their investment in their environmental credentials, they're also, by coincidence or by consequence, enhances their brand desirability. So it works for their brand. Next slide, please. So it encourages their customers, because increasingly the customer base for premium luxury products demands high levels of stewardship. And this helps them to cement their loyalty of their existing customers, but also to attract new fans around the world. Next slide, please. And the distillery, as I said, is very much based in the Highlands. It's on the banks of the Dornoch Firth, and the Dornoch Firth is actually a special area of conservation, a special protection area, and a site of special scientific interest. It has all the badges. It's a marine protected area, and it's a site that's regarded widely as being one of the least industrialised firths. That's a marine inlet in Scotland. Next slide, please. And the Dornoch Firth already has, of course, many lovely marine life organisms that are in the Firth, as illustrated on the left-hand side here. And the oysters, or rather, when we checked out the existence of oysters, there wasn't any known information about oysters in the Dornoch Firth when we started this project. Next slide, please. Next slide, please. So the big picture, zooming out to the big picture, is that around the world, oyster reefs have become extinct. They're regarded as one of, if not the most imperiled marine habitat on Earth. This information here comes from a Nature Conservancy publication in 2011. So 85% of all oyster reefs around the world are extinct. And what you're looking at there, the red and the brown areas are where things are particularly bad. Right, next slide, please. And the reason you might not have heard this story before is because many of our oyster beds in the northeast Atlantic were fished out in the 1800s. So this piscatorial atlas, as it's called, in the right-hand side here, is by Olsen in the mid-1800s. And those orangey areas are where there used to be abundant oysters that are no longer there. So there was a very large oyster bed that extended out from the Dutch and German Waddenzee out to the Dogger Bank, and there were fringing oyster reefs along the east coast of the UK into the English Channel. And this dotted area here around Edinburgh, the big dotted area there, is the Firth of Forth, which is where there used to be an oyster bed the same size as the size of the city of Edinburgh today. And what I'm going to be talking about mostly today is this smaller dotted area up in the northeast of Scotland, which is the Dornoch Firth. Next slide, please. And our initial foundation research really was investigating the environmental history of the Dornoch Firth. So this publication in Biological Conservation, we searched all the way through all the extant information we could lay our hands on, from archaeological records through to mapped sources, fisheries landings, gentleman archaeology records from the Victorian era. And we also collected shell material in the Dornoch Firth and carbon dated it onto this timeline. And what this shows you overall is that there were oysters in the Dornoch Firth since the last glaciation, so all the way back to the Mesolithic period. So 8,000 to 10,000 years they were in that system, all the way up to the 1800s, and then we have no more records of them after that. Next slide, please.
So why restore? What is the business case for restoring an oyster reef? It's not just about restoring diversity or a singular species, in our case, the native oyster. It's about restoring the habitat and the ecosystem and the species that underpins that, that's a degraded keystone species. The loss of the habitat has also led to the loss of ecosystem services. And through restoration, this provides an opportunity to enhance ecosystem services, both provisioning and regulating, to supporting and cultural. Shellfish reefs are indeed biodiversity hotspots. This links to more fish, which in turn gives and sustains access to commercial fisheries, underpinning sustainable livelihoods. They have a role in enhancing water quality through filtration and feeding, and in turn, carbon stabilization through the growth of shells, but also the deposition of sediment from the water column, and how this is integrated into a reef over time as the reef grows. To begin with, I'm going to take you through some examples that are based on horse mussel reef, which is a more widely known species here in the UK. This reef in particular is in North Wales on the Pen Llyn, and this work that I'm about to demonstrate started, and the reef is associated with a commercial fishery that is for whelks. Through potting, this study demonstrated that across 4 sites, over 50% of the catch was from the horse mussel reef alone. Not only were there more whelks, but what you can see in the graph in the bottom right-hand corner was that there were smaller whelks, indicating this area is an important nursery habitat, and it supports the early years of the whelk population. This is likely linked to the enhanced structure of a reef, providing shelter, protection from predators, and also feeding opportunities for the young whelks. Furthermore, as I mentioned, there's a commercial fishery associated with this reef for the whelks. This supports rural communities in the area. It employs 12 and has done for the past 25 years, bringing in that important economy. Now, moving on to a larger scale across the UK, through surveys of species on several different reefs. I give you an example of counts of starfish, queen scallops, whelks, and also spider crabs. As you can see here, on the reef site, when compared to other habitats, such as sand or cobble, the abundance is higher, and many of these are key commercial species. My own work focuses on shellfish and carbon. In particular, I study 2 species, the native oyster as well as the blue mussel, both of which we have slowly being restored for the oyster in the Dornoch Firth. Both species are filter feeders. They take up food from the water column, digest it, and deposit this down onto the seabed, essentially shellfish poo, or feces and pseudofeces. Both of these contain carbon. And this links into enhancing sedimentation rates through this biodeposition, but also through the structure of a reef. It's been shown with some of the American oyster habitats that this reef structure also has a role to play in enhancing passive deposition from the water column. There's a question mark here because this is something that we are actively researching in the group at the moment. As bivalve shellfish, they also grow shells, which are made of calcium carbonate. And over time, this shell and the sediment deposits, both containing carbon, become buried within the reef as it grows. Again, you'll see a question mark. This is something we're actively working to quantify, the rates of burial, but also the rates of carbon loss, which I'll come on to in a later slide. For now, I'm going to take you through a study that will soon be in publication. And this was based at St. Alp's Marine Station on the east coast of Scotland. The aim of this study was to quantify the deposition rate of the native oyster. In order to do this, we had both live oysters to look at feeding and also dead oysters to look at the shell and how the shell influences deposition, so that passive deposition I spoke of earlier. Having these two enabled us to untangle the different modes of deposition from each other. And as a result, what we were able to demonstrate is that indeed the native oyster enhances the link between the water and the seabed, tripling the rate of deposition. Furthermore, the rate of carbon drawdown was also tripled. Interestingly, what you can see here is that the shell didn't have much of an effect. And I'll come back to that later. I give you some numbers, which I'm going to take forward into this presentation. 35 milligrams per oyster per day. That was the total particulate matter deposition that was estimated through the study. This translated 1.6 milligrams of organic carbon and roughly 0.9 milligrams of inorganic carbon. These kind of numbers are really useful when we're starting to try and either understand what we've lost through the degradation of a reef, or what we may gain by restoring a reef. So moving again out to the bigger picture, see the piscatorial atlas that Bill demonstrated earlier. The Firth of Forth bed was 31,000 hectares in size, and as Bill mentioned, has subsequently been lost, including the services that were with it. At its peak, the population density is unknown, and we do have records of high catch rates at this peak period, indicating a high population, and it has now been extinct for over 100 years. So one of the burning questions for the restoration community is, what did the density of these populations look like? So to give some estimates to allow us to scale the potential carbon stabilization that may have been associated with this reef. To start, a 0.2 per meter squared as a low density estimate would give us 62 million oysters over that area, or 37 tons of organic carbon per year. Now, if we scale that up, obviously the carbon deposition will scale with it. So 50, which would be a high density reef or higher density per meter squared, is 9 kilotons of organic carbon per year. And this is a useful tool that's species specific for the native oyster. The deep oysters, 4 million over 40 hectares, which translates to 51 tons of sediment and potentially 1,300 kilograms of inorganic carbon through deposition and 2,400 kilograms of organic carbon. And these numbers allow us to revisit that other 5% that Bill mentioned earlier and start to begin to understand how the restoration of the reef relates to that remaining 5% of the distillery discharge. But it's not as simple as that. You can't just measure the shell and you can't just measure the sediment. To be able to understand whether a reef is a carbon sink, drawing down carbon, or a carbon source, releasing carbon, you must understand the balance and the routes of loss and gain. An example of a route of gain would be the biodeposition, but then again, carbon is lost over time through erosion. And all of these factors have their own variability that is related to season, is related to species, as I mentioned, and also age of the reef, location of the reef, and these must all be taken into account potentially for individual areas. But what is key to remember is that over time, this shell and this sediment containing carbon is building up within a reef as it grows. And that's what you can see here in the bottom right-hand side, is that horse mussel reef from the Penguin and a side scan of the sediments below. And with that, I'm going to hand back to Bill.
Okay, thank you, Hannah. So going into the whole of the DEEP project now, this schematic that you see here shows the Dornoch Firth sweeping through the middle of the picture and the distillery center right. And the distillery, of course, takes barley and distills that, and inevitably there's some organic material left over at the end of the distillation process. As I explained earlier, that organic material then passes into an anaerobic digestion plant now, where 95% of that organic material is digested to produce biogas. And that biogas now can be reused and is reused to fuel the stills in the distillery. So a nice circular economy story. There is 5% of that material that's left over at the end, which is well within discharge limits, legal discharge limits. But that continues to go into the Dornoch Firth, as it has done for decades. And we believe that this restoration of 40 hectares of oyster reef will account for that final 5%, improving water quality further, and then also taking some of the nitrogen and carbon out of the water. Now, it sounds simple to start with when you think, I think I'll do some oyster restoration, I'll put some oysters in the water. But very quickly you get into this process of thinking, okay, well, how many oysters will it be? And we've arrived, and I won't be able to explain in the time available how we arrived at this 40 hectares. But basically, if we're going to restore 40 hectares of oyster reef, we're going to need some shell material, because over the last 100 years that these oysters haven't been there, that material has dispersed and eroded and is no longer in the system. And we can get this material for recreating the shell habitat from waste shell, from shell processors. So we have to source waste shell material to make this work. And also along the bottom here, what you're looking at is the supply chain for native oysters as well. We have been putting 10-gram oysters into the Dornoch Firth as part of our trials because we believe that they're large enough to escape most of the predation. And if we're going to do that, we need quite a lot of oysters. We need 4 million oysters. And early on in the project, we tried to source oysters, and it took us a whole year to find 20,000 for that particular stage of the project. So to get 4 million oysters in the water clearly means that we have to energize the supply chain. So we now have to engage with the supply chain at the hatchery level or the spatting ponds. And then we have to engage and develop the work with our partners in the supply chain to on-grow the oysters to a suitable size. And all the way along here, there's innovation to be developed in how to restore oysters, how to recreate that habitat type, exactly which oyster size works best, etc. And there's biosecurity associated with translocations as well, and a whole knowledge export economy that develops out of that. Next slide, please. Okay, so there are a number of diverse challenges that start to appear when we start to embark on a proper restoration project. The first one, and the obvious one, I guess, is resourcing, particularly if you're leading the field in these kind of areas. Inevitably, a slow-growing species that takes a while to build up its habitat is going to be quite a long process. And we estimate that restoration is sort of decadal in duration. And for that reason, it's important to have the appropriate resources to be able to move forwards. And we're very, very fortunate in that we have a partner, Glenmorangie, who has been very supportive throughout this process. And one reason that they can be supportive through this process is because they're a company that can take a longer view. Their minimum product duration is 10 years. It takes them 10 years to produce their most popular Glenmorangie original. So they're used to thinking in decadal time spans rather than companies with faster turnovers that perhaps can only think a year ahead. And as I mentioned before, there's embedded thinking in their sustainability as well. So their brand is very much linked into their sense of place. So it works very well for us. But resourcing is quite an important challenge. Next slide, please. Next slide, please. The supply chain that I indicated earlier is also another challenge that we have to engage with. We need 4 million oysters, and in the rural parts of Scotland where these oysters are grown, there are very few producers who are producing anything more than a few tens of thousands. So there's an opportunity really here to develop rural enterprise and the gross value added that goes with that. Next slide, please. There's also challenges and opportunities to develop circular economy around shell waste. So shell waste comes from the processing of shellfish such as scallops. And historically, this has been an animal waste product, so classified as animal waste product category 3 under the UK legislation. And this means that it's a tricky thing to deal with. But we need waste shell, and we need about 4,000 cubic meters or more of that in order to recreate the shell material. So what we've ended up doing in this project is we've been working very closely with UK Water, with Peter Murphy, who's got right to the bottom of the standards for animal waste, and sourced shell material that previously people may have been a little bit embarrassed about these piles of shell. And we found a new circular economy opportunity for these now, of course, and we've found new standards that government can agree to for its reuse and putting it back in the sea. Next slide, please. And some of the other research work that's been going on around deep, this is Ana Rodriguez's work. When you start thinking about putting a reef in and putting putting oysters onto it, we then have to start thinking about how to make the larval oysters, the waterborne planktonic larval oysters, settle out onto this new habitat. And the way to do this, or one of the ways to do this, Anna has found, is that by putting other oysters down there, you stimulate the settlement of the oyster larvae. So all of these little graphs here represent a series of options that larvae were given in an aquarium situation. And the thing that stimulates them to settle more than anything else is the presence of other oysters. Next slide, please. Likewise, this graph you see on the right-hand side shows that behaviorally, the oyster larvae are actually spending an awful lot of time near to the seabed. And that affects the way they might disperse in the wild from native oyster beds. So, of course, one of the important questions to ask about restoring an oyster population is, does it become self-sustaining? Do the oyster larvae in the water column get dispersed over a wider distance, or do they find their way back to the natal bed? And what this research is beginning to indicate is that larvae may actually be programmed in their behaviors to try and stay fairly local on the beds where they were released from. And we're in the middle now of validating these kind of behavioral observations in the field as well. Next slide, please. So is there a rural economic opportunity? I mentioned earlier how we needed to work with supply chain. But since we've been developing the DEEP project in the last 5 or 6 years, there is now more than 17 projects across Europe, across the Atlantic Europe, that are now embarking on oyster restoration, native oyster restoration. And we were lucky enough to host the Native Oyster Restoration Alliance conference in Edinburgh a year ago. And during this conference, 155 people came from 15 different countries to share their learning about oyster restoration. And in the preparation for that, I took the liberty of polling some of these projects to find out how many oysters they needed to fulfill their targets for their restoration projects. And based on the responses, I was able to estimate that there is potentially 50 million oysters required over the next 5 years across this area. Next slide, please. And in the process, in the theme of diverse challenges, we've also embarked on a program biosecurity, because, of course, what I've discussed and illustrated to some extent is that in order to do the restoration work, we need to move oysters between growing sites and into restoration sites. And this is a translocation. And of course, shellfish aquaculture has historically been one of the main vectors for the movement of invasive non-native species, but also shellfish diseases are a major issue. So we've had to invest quite a lot of effort in developing best practice for moving oysters in a way that is biosecure, or as biosecure as possible. And one of the challenges, the many challenges that we've uncovered in the process of doing this, is that, of course, restoration has a very different risk profile to the table market for oysters. So if you're taking oysters out of the water in one place and taking them to the market to be eaten, that's one thing. But if you're putting them back in the sea, you're magnifying the disease transfer risks. And for this reason, we've realized that we need to start being able to screen in different ways using more advanced tools. And we've just started a project looking to develop eDNA tools for screening for invasive non-native species with Aberdeen University. Next slide, please. Furthermore, one of the big challenges that we've kind of illustrated earlier is that these habitat types don't exist anymore. So knowing what a native oyster reef should look like is very difficult, because there are very few examples left now. So in terms of understanding what a biodiversity target should look like, or what a restored reef will look like when it's completed, we're having to embark on a program of work which will start this autumn with Naomi Kennan doing some work with Heriot Watt University on the biodiversity of oyster reefs. Next slide, please. And also, by creating these habitats, we have to be able to optimize the process of creating habitats. We have to know the right type of shell to use, the right time to put it in the water. So we've been doing a lot of intensive work in Loch Ryan, which I'll speak about in a minute, last year with St Abbs Marine Station, looking at the distribution and intensity of oyster larvae in the water throughout the year. And the purpose of this is to be able to understand what triggers we should look for environmentally as the cue to put shell material into the water, and therefore to optimize the process of relaying shell material for oysters to settle onto. Next slide, please. So what I'm going to do now is just wind things up by giving you an overview of some of the more recent work that we've been doing in Loch Ryan in association with the Loch Ryan Fishery Company. And this illustrates some of the diverse areas of research that we're now getting into in order to inform the Dornoch Environmental Enhancement Project.
The ancient waters of Loch Ryan near Stranraer on the west coast of Scotland hold much in common with the Dornoch Firth around Glenmorangie Distillery, where the Deep Partnership has created a groundbreaking initiative to restore native European oysters to the Dornoch as part of a wider commitment to water quality and the enhancement of the marine environment. In 1701, the Wallace family were given the oyster rights to Loch Ryan by King William III, and in 1791 Loch Ryan was recorded as being rich in oysters. This bank abounds with oysters of a most excellent flavour. They are found indeed all around the shores and might be got in great quantities. The mantle of looking after Loch Ryan's oysters is still with the Wallace family to this day in the form of the Loch Ryan Oyster Fishery Company in partnership with Tristan Hugh Jones and his family, who are working to restore this ancient oyster fishery back to full production.
Loch Ryan is the last remaining wild fishery for native oysters in Scotland, so it offers us a window on the past, but it also offers us a view to the future of what the Dornoch will look like when we've completed the restoration work. But what we're interested in here in Loch Ryan is culch. What that is shell material that you put onto the seabed to enhance the settlement of oysters and create more habitat for them to live on. If we can optimise that process here, then we can enhance the last remaining oyster fishery in Scotland, but we can also work out how to make that work in a restoration context in the Dornoch. We're working to establish when the best time to put culch in the water is. And the best time is when the oyster larvae are ready to settle and when they're in the right place in the water column. And that's why we're studying oyster larval behaviour here as well. We've been diving at several places in the loch, and I've seen more oysters than I've ever seen in my life. There are rather a lot of them out there, and in some places they're really dense, and we're beginning to see the habitat types that have long been lost. Oysters are really important because they create habitat for other species, which can include commercially important species, but they also clear the water of particles, so they improve water quality, and they deposit sediments onto the seabed and lock away carbon.
I'm looking at how oysters facilitate the capture of carbon, so how they facilitate the downfall of carbon, which can be sediment, but also in their shells.
Increasingly, carbon stores are becoming very important to manage as we go forward.
Deep has been a catalyst for action to restore the native oyster for nature, business and communities right across Scotland. This study of one of the last living wild oyster fisheries in Europe also inspires our vision for what the Dornoch Firth could look like in years to come. We are very grateful for the support of the Loch Ryan Oyster Fishery Company and Scottish Enterprise in helping us develop this important partnership.
Okay, thank you very much. We'll stop at that point. That's a suitable summary of our presentation, I think, and we'll accept some questions.
Excellent, Bill and Hannah. There were some lovely videos in that presentation. Thank you very much for today's talk. To all our participants, now is the time that we are moving on to the Q&A. So if you have a question that you would like to ask, please pop it in that box that is at the bottom of your screen. I'm just going to put out a link into the chat to all of you. That is a document that contains all those links that you just saw on the very final slide that Bill had was talking about and Hannah was sharing on there. So Bill and Hannah, do you mind just talking about what those links are, just whilst people submit their questions, maybe what's in that document there for them?
That's fine, yes. So there's a number of there's a number of links there to some of the movies that we've shown, and also some other ones, and the Dornoch Environmental Enhancement Project site. We've also got a number of publications which we mentioned in passing, and some of them are open access as well. So they should be picked up by those links as well.
Okay, thank you. Also, I learned a new word today, that culture word. That was good to know. And we are now going to move on to our first question, which is coming in through the Q&A. So for all our attendees, pop it in there. And so our first question is, where did the oysters that went into the Dornoch Firth come from? So you mentioned that there were 4 million oysters that you needed, perhaps a huge amount. So a bit more elaboration on where did you get that massive supply from?
Well, we haven't completed the restoration yet. We're on our 20,000 oyster stage at the moment. And that means that our first brave 300 oysters actually came from Loch Ryan. They were our first test oysters, and they grew and survived very well for 2 years. And then what we did is we started to lay down oysters at 10 grams that came from aquaculture. Some of them came from Loch Nell on the west coast by Oban, and some of them came from up in the northeast as well. Sorry, northwest of Scotland, from Patrick Blow's site there.
Yeah,
and sorry, more recently from Little Loch Broom as well, from Moorbeck oysters.
That's great to know, a large scale kind of collaboration on
That's
getting this project on the go. So our second question then asks, the high larval settlement seemed to be induced by the presence of spat. Did you test large oysters and oyster shells too?
Yes, we tested shell material from an oyster bed, shell material from that wasn't from an oyster bed, and we also tested rocks from an oyster bed and rocks from that weren't from an oyster bed, and the biofilms. So we believe the biofilms associated with material coming from an oyster habitat was also provoking them to settle a little bit more. But the thing that provoked them the most was the presence of another oyster spat. We didn't test them. Looking at Christine's question, we didn't test adult oysters in this particular experiment. We tested the spat, but looking at the rest of the literature, it seems that it's likely that it's the presence of oysters that's actually triggering that.
Mm-hmm. And what do you think, in your opinion, how long before the Dornoch is likely to be sustainable for commercial source of oysters?
Well, I get asked that a lot. So actually the Dornoch Firth has 2 conservation orders which prevent the extraction of any species apart from the blue mussel, for which there is an ancient and historic right for tame people to fish them. So the short answer is no, there won't be any commercially available Dornoch oysters. However, from the start, we've been very enthusiastic about the idea that inevitably the spat oysters from this population will spill out into the wider Moray Firth area. So it's entirely possible that we may inadvertently trigger settlement elsewhere, which will lead to a commercial fishery. But what I'd personally like to see is a network of sites that develop across that wider geographical area. And of course, restoration for conservation is one business model for restoration. Restoration for carbon mitigation that we alluded to in our talk today is another business model, but restoration for fisheries is another equally valid business model for restoration.
Sounds good, thank you. David also asks, where does culch, or was it clutch or culch?
Culch.
Where does that come from?
So shell culch at the moment, what we would love to be able to use is oyster shell because it's thick and heavy when the animals get big. But of course, you know, inevitably that isn't available. It certainly isn't available in the quantities that we need. So we have to be pragmatic, and we go to shell processors for our shell culch. And that means that we can either get culch, which is blue mussel shell from shell processors, or more commonly scallop shell. And therefore, we have 2 or 3 shell processors that we've been working with across Scotland that can supply the sort of volumes that we're after.
Yes, a really nice demonstration of that circular economy that you mentioned right at the beginning, and then kind of brought up again throughout the entire process, which has been a really key part of this project. Our next question from Ben asks, what are the main parameters you measure to assess the ecosystem services, e.g. nutrients, productivity?
Okay, well maybe Hannah would like to answer about the ecosystem services she's been working on.
So in terms of carbon, for the biodeposition, at the moment we've been measuring the rate of carbon down draw by oysters. What I presented there was work from St. Abbs Marine Station. So that is to give us the first look at quantifying that provision of carbon stabilisation. Further to that, we are also collaborating and running samples over at SAMS to look at the respiration calcification side of things. My work does go further with blue mussel side of the project, actually working on blue mussel beds in Scotland to estimate how much carbon is in the beds themselves. We're looking to take that biodeposition study from St. Abbs into the field as well, so we can actually quantify on-site rates of deposition. And as I mentioned, that's quite variable. So it's important if you're able to get the numbers across the spectrum, potentially of the sizes of oysters, but also of the seasonality, because it will change.
Understood, thank you very much. Phil then asks, our next question is the overall goal of the project purely to enhance biodiversity, or is there an aim to support a fishery in the future? So Bill, you've already kind of said that if there's additional spillover that a commercial fishery could kind of form on, that would be a great additional result, I think.
That's right, yeah. So it's primarily a biodiversity enhancement project, but we have recognised that there's an additional goal, which is about the carbon capture and the water quality improvements that this project generates.
Mm-hmm. Our next question comes from Louise, who is down in Cornwall, where they have a native oyster fishery as well, and is still performing, but only by sailing vessels. They've experienced issues with invasive Pacific oysters as well at their location, and they're trying to manage them through oyster bashing, but it's an ongoing battle, they say. Have you had any issues with these Pacific oysters? And if not, what do you think might be, how are they evading the invasion at the minute with these invasive species?
Okay, well, the history of the Pacific oyster establishment in the UK was centred around the south and west of the UK. They are spreading through our coastal waters. And at the moment in the northeast of Scotland, in the Dornoch Firth, we don't have the Pacific oyster at all. I know that they can, they are, first of all, commercially, they're really important to the aquaculture industry, so I fully recognise that. The other thing is that given that we don't have it in the Dornoch Firth, we're not in a hurry to introduce, in fact we're not going to introduce the invasive Pacific oyster. So we actually spend a lot of time screening them out of our supply chain and making sure that we don't have them. And the eDNA project that I referred to earlier, one of the primary objectives of that project is to be able to detect the presence of Pacific oysters, because of course if you've got a mixed bag coming from an aquaculture site, you may not be able to visually tell that you've got a 10-gram Pacific oyster there, but you can tell genetically if there's some in your tank of 1,000. So it is a potential complication that we recognise, and we're avoiding it at the moment. And I know that other restoration projects have a different relationship with the Pacific oyster.
Our next question, thank you. Our next question kind of builds on that about biosecurity. Could you tell us what the threats have been to the remaining fishery in Loch Ryan over the years and how these have been managed?
Well, I don't actually, I've not had a long enough involvement with Loch Ryan to know what the historic threats to the fishery have been. I do know that from the work that we've done that there are, as there are in many places, there are invasive species present in Loch Ryan. In many other places there are as well. So there's things like Stylochlava, for example, which is a stalked sea squirt. And we've, from our own surveys, we've seen 6 different invasive species in Loch Ryan. And from our trials and our biosecurity, which we haven't published yet, we will do, we've tested our translocation methods and our biosecurity processes, and we've had 0 transfer of those species to the Dornoch Firth through our closed facilities. So we call it our biosecurity firewall. So we've been quite successful so far at preventing moving these problems around. But in terms of the Loch Ryan fishery, I don't know what the threats have been over the years. But there is a population there still. It's the last remaining one in Scotland, and it's been fished since 1701. So it's quite a good news story, really, in many ways.
Absolutely. Our next question didn't come in through the Q&A, it came in through the chat. If anyone, any of our attendees want to ask a question, please pop it in the Q&A box. But this question asks, well, it says, moving larger oysters between water bodies is one way to source restoration stock. One of them, another alternative is newly settled spat from hatcheries. What is the prognosis for hatchery product for restoration, do you think?
Okay, so there's a number of different ways to crack this nut, one of which is to on-grow them in other sites and then translocate them in, which is what we've been doing so far. Taking spat directly from the hatchery and growing them on in the site is another way of doing it. That removes a lot of the biosecurity risks, particularly if you've got a good clean product coming from the hatchery. But it does mean then you have to embark on the process of essentially farming them in your site as well, which means that you then have to get into all the associated infrastructure, licensing, etc. So when we embarked on this 6 years ago, we recognized all the main ways of doing it, and we always imagined a very mixed approach to the whole restoration process, such that whichever started to work the best first, we would hitch our wagon to that particular approach. So far, we've been growing them to 10 grams and then putting them in. But equally, we could embark on taking spat directly into the system. Watch this space.
So yeah, moving on to our next question, which I expect is a question for Hannah. Why do you think that there is little difference in sediment deposition between the dead oysters and having no oysters in the St. Abbs experiment?
I think that's a great question. So as I mentioned, that's something that I'm taking forward currently with my research. I think what it stems from is the way the oysters were laid out in the experiment was with very low relief. So over a gradient of complexity, I would expect that we would see obviously an increase in bottom roughness, which would in turn increase that deposition. So this is something that we're working on at the moment within Heriot Watt. I hope that answers the question.
I think it does, yeah. Our next question is also for you and regards microplastics, which are a huge issue. Do the oyster feces and pseudofeces help deal with this biodeposition and potential plastic sequestration?
So that's a great question there, Dave. I don't focus on looking for microplastics within my samples, so it's not something I can completely comment on. But I would expect through the integration of the structure of the reef, and I know there is some work that's come out recently on this in terms of that capture of plastic within to a bed itself. I think the work's on blue mussels.
Okay, thank you. Our next question from Phil asks, are you seeing success from the initial 20,000 oysters that were deposited in terms of survival, growth and reproduction? Is there any data that you're already getting feedback from on that first laying of 20,000?
Yes, we have been getting very good growth and survival. I was really hoping to have some great numbers from this year, and we were due to be out in March, but of course lockdown has completely flummoxed that. So yes, we do have good growth and survival, particularly in the east of our site, and I could show you pictures of beautiful growth rings on shells from last year's season. So yes, we are getting good growth and survival, but we're hoping to be able to validate that and produce some proper data on it fairly soon.
I am just noting the time, and I'm aware that we are scheduled to end at 2 o'clock. So I hope we have one more time for one more question, which I'm going to take from the most recent question that was put in the Q&A box. And it says, 2 quick questions. They've actually written that. In the video, the oysters seem to be laid directly onto the culch with no protection. Are you monitoring the bottom currents and siltation? Are they a potential issue? Dispersing the loose oysters away from the sites or being smothered? And any idea of how they're doing RE predation?
Yep, so that's Jose that said that, right? Okay, great. Hello, Jose. Yes, when we initially embarked on this, our first of our 20,000 oyster stage, we laid them directly onto the scallop shell, and it sounds a bit daft now to say it like this, but you know, we've since realised that, of course, with the tidal currents that we have in the Dornoch Firth, that there was going to be a reasonable amount of movement of things. But what we have found is that the scallop shell in particular seems to imbricate itself, which I think is what geologists call it. The shells start to stack up in the tide, and they start to infill with small amounts of sand. So what you do find, what we do now is we put the shell material down first, we allow it a few months to stabilize, and then we put the oysters down onto a much more stable surface. So those video images aren't that representative of what we do now. We've learnt along the way, and we have a more robust way of doing it. So yes, they stay put much more when you let the shell settle first.
Okay, thank you very much. I have one last question, and it's for Hannah, from Mark. In terms of total carbon budget, are you taking into account the biochemical balance of sequestration as the creation of shell material creates a net increase of CO2 release?
That's a great question, and that is something that we're working on at the moment. And as I mentioned, it's not as straightforward as some work previously may suggest of just measuring shell as how much is there, or just measuring sediment. You really have to take into account, as you said, the actual equation, the balance of calcification, respiration, the deposition, and then outside factors, which is all what I showed in that diagram, and hopefully that paper will be out within the summer.
That's great to know, and I think that's a really good point to end our webinar today. So thank you very much, Bill and Hannah, for being our first duo MASTS webinar. I think that worked really well, and some lovely videos that you showed us as well. This session for all our attendees was recorded and will be uploaded to our YouTube page at the end of today. Thank you very much, Bill and Hannah, again.
Thank you.
Thank you.